So the next 45 minutes, I will be talking about my life with a heavy focus on my education and research experience. I will be talking about how I became involved in the research that I do specifically on the brain and a lot of what I've been doing, well, for a while now. I was born and raised in Tacoma Park, Maryland, near Washington, D.C. You can see various pictures here of me and my family. I have a twin sister. I am deaf. My sister is hearing. My parents did start learning ASL when I was born, and they found out I was deaf. I'm very fortunate that they took the effort, made the effort to take sign language classes and learn to communicate with me. I was able to sign with my parents and my family at home. You can see a couple of my baby pictures. I was real cute. I wanted to share some of those. I had complete full language access for my elementary and school education. I went first to the Kendall Demonstration Elementary School for the Deaf and then the Model Secondary School for the Deaf. From ECE to kindergarten, I went to school with my sister, both me as a hearing person, a deaf person, and she as a hearing person, went to the same school, but then elementary school she went to, public school, and I went to a deaf school. So for the first 15 years of my life, I was learning and communicating in sign language everywhere that I was. After I graduated high school, I thought about going to Gallaudet or RIT NTID or a different college. Well, my mom worked at Gallaudet at that time, so I wanted a different experience. RIT was in too cold of a location, so I didn't want to go there. And so I chose kind of a random college, the Randolph-Macon College, which is where I went for my undergraduate experience. It was my first experience at a hearing environment, in a hearing environment. I went from a fully deaf environment to a fully hearing environment, which was quite a challenge and a learning experience for me. I had to learn how to use interpreters for the first time. I had to learn how to advocate for myself, which was a tough experience. I used ASL interpreters for my classes, but not for extracurricular activities. The clubs and other activities I was involved in, I just would text or write back and forth or communicate on my own the way I could. It was very frustrating for 4 years, but I did get through it, graduated with my undergraduate degree. I was interested in psychology and to begin with, clinical psychology. But then I actually was more interested in if something were to happen to your brain in some way, what would the impacts be or what changes would happen? And so, if you break a bone, when you break a bone, you can get an x-ray or something and see that it's broken and fix it pretty simply, fairly simply. But often when you have some effect on your brain, it's difficult to find out the cause or what is wrong and how to fix it. The brain is something unique, and I wanted to learn more about it. So I realized that clinical psychology was not for me, and that I really wanted to focus on neuroscience and how the brain works, how everything, the neurons and everything in the brain work together. That's really what I wanted to study. So I wanted psychology and neuroscience. Now, Randolph-Macon had biological neuropsychology, but not cognitive neuropsychology programs. The difference in those are that cognitive neuropsychology or cognitive neuroscience focuses on language and the development of language and other aspects. And then the other type is focusing on behaviors and how your body functions based on your brain synapses. So similar to rats, when you put them in a maze and you look at what behaviors they elicit based on different things, that's that field, which is not exactly what I wanted to study. That's behavioral neuroscience. So for my undergraduate research, I worked with Dr. Kelly Lambert. You can see her picture in the top right of the slide. She helped me dive into neuroscience and that area. She used looking at rats as a model for depression. And if a rat becomes pregnant, the brain becomes resistant, or wanted to see if there was a resistance to depression or anxiety. So they looked at rats to see if they could compare that with humans. So I worked with her for 2 years. I spent hours and hours looking at pictures just like this that you see on the top of the top pictures. The red dots represent neurons. My job was to count the red dots, literally count them. 1, 2, 3, 4, 5, 6, 7. Then move on to the next one and count how many red dots. That was my job for 2 years, counting neurons one by one. So much fun. So behavioral neuroscience work was with rats and mice, and then I realized I could do some work in a different area. I didn't like the animal experiments side of it. I felt bad working with the animals in such a way because we might expose them to certain environments and then we would to be able to look at their brain and analyze what was happening, we would have to kill the mice or the rat and dissect the brain. I didn't like that at all. So then I shifted my focus to humans and experimentation with humans and the human brain. This was more cognitive neuroscience. So for this, I developed a senior research project for me to better understand the brain. I wanted to look at hearing and deaf people and compare them, specifically looking at a study done by Dr. Matt Dye and Dr. Peter Hauser and Dr. Daphne Bavelier. So they tested individuals' ability to look at something in the middle of a screen and see if they could also catch something in their periphery at the same time. In this study that they found that deaf people have a broader periphery than hearing people and can catch those images in the periphery better than hearing people can. So for my senior thesis experiment, I wanted to see if there was a difference in deaf and hearing individuals. So we did a specific training. So for my experiment, we gave deaf and hearing people a test on the UFOV, the Universal Field of Vision, field of view. So we gave them this test, and then we trained each group of participants on that same test. After a few weeks, we tested the participants again to see if there was an improvement. We wanted to see if the deaf individuals would have a faster rate of improvement than the hearing people, or if there would be the same results. So that was my first project. It wasn't real successful. The biggest reason was because my participants did not do the training. So this project was a bit of a fail, but it was good enough for me to satisfy my graduating requirements. So at the time that I did my senior thesis project, I didn't know who doctors Matt Dye and Peter Hauser were. But after my bachelor's graduation, I reached out to both doctors Dye and Hauser and asked if they might be looking for someone to join their lab. And they were. So I applied and was accepted to join this project, which then led to my post-baccalaureate work.
So for my post-bac work, I did a 3-year research associate job working with Dr. Matt Dye and Dr. Peter Hauser. They hired me to study the development of attention skills in 125 deaf children. So what I did was travel around the country for 2 to 3 years doing this testing with deaf kids all across the country. My research question, our research question, was looking at deafness and sign language experience and how that impacts temporal attention skills in deaf children. The reason that we're interested in this is because there is previous research by a different group that tested deaf children's temporal attention skills. And let me explain that temporal attention skills means the ability for someone to look at something over time and see it as it changes and be able to notice those changes and remember them. So that's temporal attention, which is a little bit different than spatial attention. And that other research group found that deaf children had reduced temporal attention skills compared to hearing children. So their hypothesis on why that happens, according to this other research group, is that because deaf people can't hear, therefore they're missing that auditory sound input. And so they lack the ability to develop temporal attention because they lack an ability to sequence and count sequences. So the idea is that because deaf children can't hear, they can't develop those temporal attention skills. Our research group questioned those findings, whether it's because of deafness or potentially because of other reasons related to language development or communication, for example. So we tested deaf signing children over 3 years to see what their development looks like. And we published 2 or 3 different papers from that research study. In summary, what we found is that signed and written language skills do predict attention skills in deaf children. And signing deaf children perform equally to hearing children in terms of temporal attention skills. So we were able to show with our research that for deaf signing children, their deafness is not going to impact their temporal attention skills. What's more important is language and communication access. So that was a great experience for me. Thank you to Matt Dye and Peter Hauser for that experience. And that job really helped me understand how to work with deaf children, how to do psychological experiments like that at a larger scale, how to run a big project like that, for example. I learned a lot of really valuable skills. I became more interested in the brain, and I knew that I wanted to do cognitive neuroscience research. As an undergraduate, I did more behavioral neuroscience. My bachelor's degree was more in psychology and cognitive science, but I had not yet done imaging or MRI brain research. So when I started my PhD, I did my PhD in San Diego at, this is a long name, it's SDSU UCSD, Joint Doctoral Program in Language and Communication Disorders. So very long title of my PhD program. But basically that program focused more broadly on language and communication disorders. Many students in that program did research on autism, aphasia, and a number of other areas. My focus was on sign language, obviously not a disorder, but it still fit within the program parameters. Luckily, this was my first time doing cognitive neuroscience research with MRI data. And I was in this program for 6 years. It was fun, and it was also very difficult. Now, I started this program in 2019, and 5 months later, of course, the COVID pandemic hit. So for the first 3 years or so of my PhD, I basically stayed home. All of my classes were remote. Most of the workshops and resources for me to learn MRI research were canceled or dropped. We weren't able to do in-person research. I had to do a lot of my learning on how to do MRI research and how to analyze MRI data online. I had to do that remotely, and I had to really teach a lot of that to myself. It was very challenging. It was a lot of fun, but it was definitely very challenging. My dissertation research, in a nutshell, looked at 3 different research questions. First, what does neural network for sign language comprehension look like? Second, how does input modality influence the neural network for language? And third, how does early language experience influence language activation in deaf signers? So now I'll walk through my dissertation research and answering those 3 questions that I did over the last 2 years. If any of you saw my dissertation defense, you'll recognize some of this content. So the neural network for language is supramodal, and the word supramodal means basically whatever, regardless of what language it is, whether it's written, sign language, or spoken language, all 3 activate the same networks. The brain doesn't care how the language is delivered or the modality of language. It's more about the connection of words and meanings, and there's a systematic relationship that triggers and activates the brain in the same ways, regardless of whether it's sign language, a visual language, an auditory language, or through touch. So previous research findings typically look primarily at spoken and written language. When you do an MRI scan for anyone, deaf or hearing, for language comprehension, how do you identify or target which regions in the brain are activated? Typically, there are 2 tasks that are used. The first task has some sort of language sentences that are shown with an actual meaning. And then the second task shows stimuli that are sounds, for example, or letters, but not actual language. There's no linguistic structure to it. There's no meaning. So the first task, the actual language, we can see how someone processes that language and understands what that sentence is saying, basically. So for example, you can see here in written English, he sat down on an old box and focused on the problem. And as a person reads that sentence in the MRI, we can see the activation in their brain correlating with them processing that sentence. And then next, you can see the pseudo sentence. There are real letters in lines and real shapes of language, but there's no actual meaning to it. So if you subtract task 2 from task 1, then we can target what the actual language networks are. For written English and for spoken language, regardless of whether it's written or spoken, we can see the same activation of the same regions in the brain. That's those 6 regions on the right side of my slide. And typically that's stronger in the left hemisphere. And again, this is the supramodal, frontal, temporal area of the brain. Supramodal frontotemporal network. For my research, I wanted to look now at sign language comprehension with those same two tasks. So this is what it looks like. So these are not actual videos in the PowerPoint, but basically it's very similar. The first, oh, can you click on it? Let's see. Click on it again. On the other one, on the first one. So there you can see Alice's video. This is Alice in Wonderland. And then the blurred one has colors and shapes, but no actual meaning. And again, if we subtract task 2 from task 1, what does the brain activation look like? This is a scan, and I scanned 15 deaf signers and did this task. I identified the regions that were activated for both tasks. And you can see here that it looks very similar. And I was thrilled. It's nice to replicate previous work with sign language. Now, with my data, seeing these regions plus here we go. We also saw some additional regions that were activated. And typically those regions are activated more with written English in previous literature. So that made us wonder what those regions are doing exactly. I wrote a review paper, which I'm very proud of. It was recently published, so feel free to find it and read it if you want. You can see the citation in the bottom left of my slide. And this is basically an overview of what is activated and what happens when people are processing sign language. So first, the colors, lines, visuals will go to the occipital region to process for the early visual processing. That information is then divided. Some of it goes through the ventral area, which processes the form, body, face, and hands. That's more specialized to be able to identify people. And then from there, it goes to another copy of that information, goes to the more dorsal area of the brain that processes movement specifically. So it goes to both of those regions simultaneously. And then from there, that information is then sent to the SMG and the STS area. Those are two regions of the brain that process visual phonology, meaning hand shapes, phonological movements in ASL, and location. And then the phonology, once that's been identified, then the area of the brain activates that's processing words, sentences, and the actual meaning of what you're seeing. So that is the sign language comprehension network. I made this picture myself and I'm very proud of it, very proud of this flowchart that shows how the information is processed. From here, I wanted to try to divide those three parcels of language processing. So the visual, the phonological, and then the actual semantic meaning. To do that, I wanted to look at baseline, movement, and form, pseudo sentences, and then actual sentences. I'll show you quickly a sentence and then a pseudo sentence and what they look like. Can you click on both of them to play them? So you can see there's a few different sentences that were shown in ASL. And it's something that you can easily watch and understand. And then can you play the pseudo sentences?
You see the pseudo sentences have ASL phonology, hand shapes, movement, and locations that will activate the phonological regions and the visual regions in the brain, but they will not activate the semantic meaning or sentential areas of the brain. So we wanted to compare that with written English. There are parallels to written English, but ASL with the videos that you see me, you see the color, hand shape, movement. But in English, you just see words that come up on the screen. And so we give the ASL sentences, the ASL pseudo sentences, English sentences, and English pseudo sentences. And we wanted to see what the brain does for each of those types of stimuli. So for example, here's one graph that I will show. There will be a lot that will pop up here, but just try to take an overall view. The bars represent the response of each region to each different stimuli. So you on the right, you see the OCC, the occipital, and then the OCC temp, occipital temporal. You'll see the orange is the ASL sentence minus the ASL pseudo sentence. And the occipital sentence responds heavily to the sentence, but it does not respond to the English sentence. It responds to the ASL signed sentence, but not the English sentence, which makes sense because ASL as a language has movement and it stimulates different areas of the brain as compared to written English, which doesn't have as much visual stimuli. The rest I will show you here shows the network, the neural network in the different regions. You see fairly clearly what is interesting about these graphs, or this graph as a whole, is the regions respond to the same to ASL and English, those on the left, meaning they respond to language stimuli. Doesn't matter if it's signed or written, doesn't matter the modality, the brain will respond, activate in the same way. Some regions, for example, the fusiform, the SPL, and the occipitotemporal regions, don't respond to either ASL or English because those areas are involved in modality-specific processing. So they don't respond to high-level language processing. That's a large part of my dissertation, actually, was showing that there's a contradiction between sentences and pseudo-sentences and where they are processed. And, based on any type of stimuli, some regions are activated versus others. One exception seems to be the occipital regions, specifically for high-level language processing of ASL. It's interesting for neuroscience because there are assumptions that these regions are only involved in visual processing. But for ASL users, there's a high level of language processing that happens, not just for the visual intake, but for processing the information itself. So it's more about the language itself and what you're receiving, not just the color and movement of the language. Second part of my dissertation, I was interested in early language experience and its effects on on neural activation for language. There are not enough, but a few studies on this, looking at the brain's response to language differences based on if you learn and acquire the language earlier or later in life. When deaf people acquire sign language later in life, meaning they were deprived of language early on, we see that their brain, well, if you'll see the frontal regions here in these diagrams, there's less activation. There's less activation in the temporal regions as well, but there's increased activation in the occipital and the parietal regions of the brain. Researchers often interpret that to mean that the frontal and temporal regions involve high level language processing. But really, it's less so because energy is needed for attending to the perception or reception of the language and the perceptual processing, which happens in the parietal, more occipital and parietal areas. If you learn language later in life, as you're intaking the sign language, you're working harder to process the language itself, the hand shape and the phonology and all of that. More energy is spent on understanding the language more than trying to analyze for meaning. So for my dissertation, I did a replication of a previous study with my data. Most studies use sign language age of acquisition, AOA, age of acquisition, as when a person learned a sign language and what age that might be, from 0 to whatever age. And then, you're supposed to pick one age and then they plug that into their model. But I didn't feel like that was sufficient. I wanted to measure any type of sign language exposure. For example, if a child learns ASL at age 8, but prior to learning it fully, they were exposed to a bit of language. Maybe their teacher used spoken language most of the time, but occasionally used sign language. They had exposure to the sign language, but they didn't learn it. Therefore, their age of acquisition would be looked at as 8. But when you had access, was maybe earlier on. So I asked participants when they were exposed to sign language versus spoken language, and they had a scale that they could choose from. And they rated, there were 4 different ratings: before 5 years old, in elementary school, in middle or high school, or as an adult. And then I weighted the ratings. If it were earlier, they got a higher weight. And if it was later in age, then they got a lower weighting. So because early language exposure is more important for the brain development as compared to later language exposure, if someone was exposed to sign language early, I gave them a higher weight than if they were exposed later in life. And then I calculated that into one score for their access for each participant. So I used statistical analysis to see the sign language access effect on brain activation, the age of sign language access. If you'll see here, this chart, if a person has less sign language access, it's the orange line varies from 0 to the higher to 1. If you have a score of 1, then that's a higher language access. So you can see the differences here. That's the darker red. So the lower score is higher or earlier language access. And we see that that activation happens in the occipital region, occipital parietal region of the brain. The occipital parietal region will have a higher activation if you have less sign language access or a lower sign language access. So this shows if there's low early access to sign language, then the phonology and semantic processing will be higher. The activation areas will be higher. That was a brief summary of part of my dissertation research. But to summarize further, the neural network for sign language comprehension is composed of two main areas, sets of regions. Modality-specific regions and the occipital and parietal regions. And those are more for analyzing sign language phonology and visual processing, but don't involve high-level language processing. It's more for comprehension of the language. And then the frontal and temporal regions of the brain respond to written English and ASL comprehension in the same way. And they don't care what those regions don't care what modality, they just want the language. An exception, again, as mentioned previously, is the occipital parietal regions. The occipital region has more activation for ASL, but not for written English. We don't know why yet that is the case. We have no idea why that is happening at this point. We just know that that is the case. Even in skilled fluent deaf signers, if they had a lower early access to language, then they have a higher activation in the occipital parietal regions. So what I'm doing now, I graduated just this past August, 3 months ago. And just this last month in October, I moved from San Diego to London. I'm currently in London. It's now 6 p.m. It's dark outside here. I moved to London because I got a 2-year fellowship from UK Royal Society to work at the University of College London with 2 researchers, Dr. Mariad McSweeney and Dr. Sam Evans. I will be looking into what type of sublexical information is represented during BSL, British Sign Language fingerspelling, and reading in deaf readers. We're looking at letters, movements, for fingerspelling, and reading text, and looking at how they overlap or how they compare. And we want to see if we can look at how to help deaf people learn reading in a different way. We are curious where in the brain this happens, and we'll be looking at that. The main idea behind this research project, I like how previous researchers have looked at fingerspelling as a bridge between sign and text. Before deaf children learn the language, they understand fingerspelling in a more overall kind of big picture way. They then build upon that to understand more and understand quicker when they are fingerspelled too. Then when they learn reading, they have to map. They have to remember as they learned their finger spelling and then they learned to parse it out. Then we look at BSL. Of course, we're going to be looking at BSL and how they look at the different letters to map to written text. Our goal is to use fMRIs to see how the brain does map fingerspelled letters over written letters. So that's what I'm doing now. Just started.